This is a question that focuses primarily on antibodies and more specifically on monoclonal antibodies and a little bit about drug trialing. So the first thing about any question that has anything to do with antibodies is that you always need to consider that an antibody is going to bind to one specific type of pathogen, because an antigen's binding site is complementary, so it's an exact fit for the antigen on the cell surface of any sort of pathogen. Right, so it starts off with a virus called RSV causes severe respiratory disease. Right, it could be any virus, it could be any pathogen. It doesn't matter if you've never heard it before. Suggests 2 precautions that a person with RSV could take to reduce the spread of the virus to other people. Right, so in this case, you need to think about what they're going to do to go and stop those routes of transmission. So not go near other people. Make sure that they're sanitizing their hands after they've been to the toilet or after they've sneezed or whatever. Put stuff up in front of their mouths. Right, lots of obvious things. Don't overcomplicate it for yourself. Right, it says one treatment of RSV, it uses monoclonal antibodies which can be injected into the patient. Suggests so scientists can produce monoclonal antibodies using mice. The first step is to inject the virus into a mouse. Describe the remaining steps of this procedure. Right, so remember that when you go and inject any sort of pathogen into any living thing, you are going to get some sort of immune response. The immune response that produces antibodies is caused by a B cell. Right, got to consider it's only worth 3 marks, so they only really want 3 stages. So we inject the mouse with the virus, like it's already said. You extract the B cells that produce antibodies to it. We want those B cells to be able to last forever, so you go and fuse those B cells with some sort of cancer cell, so they last forever. Then using that, you grow that cell in a growth medium, and that growth medium will get full up of the antibodies that that particular sort of cell is producing. So the next part goes, describe how injecting monoclonal antibodies for RSV helps the patient suffering from disease. So this is what we said about the antibodies being specific to that disease. So you need to talk about things like the antibodies will bind to the antigens on the surface of the virus. That will then go and help the immune system sit, so you'll increase the rate of phagocytosis, or that will inactivate the virus, or that will stop the virus from entering the cells. Anything like that would be fine. Right, the next part of the question all focuses on drug trialing, and drug trialing in terms of using monoclonal antibodies. So it starts off with a trial was carried out to assess the effectiveness of monoclonal antibodies to patients with RSV. Right, some patients were given a placebo. Why were some patients given a placebo? Right, remember, in all drug trialing, half of the patients will be given a placebo to make sure that people aren't cheating when giving results. To make sure that there's not something called the placebo effect, where if people take some sort of drug, whether it be real or not, the fact that they're taking a drug will make them feel better. So a number of patients had to be admitted to hospital as they became ill with RSV, right? The data is given in the table. If they're giving you data in the table, you will have to use it. The trial involved 1,500 patients. Half of the patients, group A, were given a monoclonal antibody and half of them were given a placebo. Calculate the total number of patients admitted to the hospital with RSV during the trial. Right, so if you've got a trial that involves 1,500 people, if you're looking for the percentage of them that were going to go and be admitted, you would times the total percentage, so 14.8, and you would times that by the 1,500 to give you that total. Last part, evaluate how well the data in Table 3 supports these conclusions. Right, so monoclonal antibodies are more effective at treating RSV than the placebo. The data in the table doesn't actually tell you whether it's worked or not. It only tells you the percentage of patients within each group admitted to the hospital. So the fact that it doesn't give you an exact number, 4.8% isn't that much less than 10.4%. Although you could argue it the other way around. You could say, well, far less people were admitted who had the monoclonal antibody. Right, go back, have a go at this question. Try and think your way through and try and squeeze out as many marks as possible.